I'll do a very brief overview of what Airvale does, why we take the approach we do, and what our goals are and what our outcomes to date have been, and then open it up for questions and any discussion you want to have on the topic. A couple of you, Dr. Cobbs, Dr. Delishaw, have been through this presentation before, but I do have some clinical updates for you, so I think I'd be happy to see where things are moving. I assume you all are familiar with who Dr. Lee Hood is. If you're not, Dr. Hood is a both local and national and international well-renowned, well-known scientist and entrepreneur. Dr. Hood, for those of you who don't know specifics about him, long ago sort of became one of the folks, one of the physicians and then researchers who really decided early on to focus on this area of expanding our understanding of systems biology. How do we look at multi-omic systems simultaneously to separate signal from noise and find actionable outcomes, both in the disease as well as in what now in the area of preventative care. On top of his research, which has led to both the awards such as the National Medal of Science, the Kyoto Award, the Lasker Award, he's also co-founded 15 companies, including Amgen, NanoString, North Shore Bio, and of course, probably most well known for the early work he did on the Human Genome Project, where he led one of the groups that did the first human genome sequencing and invented a lot of the first 4 instruments, in fact, that were used to actually do human genome sequencing. For years, in fact, about 20 or 30 years, Lee has worked on this idea that for most of us, we have a misconception about what drives the basics of human wellness. When you look at a series of meta-analyses in this space, what we really have come down to understanding is that as it relates to actual human wellness, the ability to keep people from becoming ill, or the ability to potentially mitigate or arrest illness in the area of chronic disease, 60% of what drives that, of course, is lifestyle and behavioral choices. Top 5 chronic diseases in the United States are pretty much well aligned with those lifestyle choices: tobacco use, alcohol use, stress, nutrition, and choice on activity or lack thereof. 30% is driven by one's genetics. And only 10% is really driven by how one actively integrates or works within healthcare on any given day in terms of one's meeting with and spending time with a physician any given period of time. This doesn't mean that the 10% doesn't matter. Of course, once a disease state starts, it quickly becomes the 90% of health, or of course in the case of accident or other. But when we look at what drives a lot of chronic conditions in the United States, which is the biggest cost driver of our healthcare system, and of course a leading cause of comorbidities, I think, in looking at your case studies, one that you spend a lot of time having to work around: obesity, people's lack of mobility, their other choices, tobacco use. One can see that lifestyle choice and its interaction with genetics often is a predictor as to whether or not someone is going to progress and stay towards a state of optimized wellness or a state of progressing towards a disease or a chronic care. Lee has been focused on this area for about 20 to 30 years, and what he calls this is P4 medicine, the idea that if we're able to use a multi-omic analysis and combine that with the right type of health alliance and health care, we can actually provide individuals with a type of health care that is personalized, predictive, participatory, and preventive. So the idea behind P4 medicine was launched because when you look at the actual the wellness industry as a whole, as we all know, it's a multi-billion-dollar industry that doesn't have much evidence behind it. And yet we also have a huge focus on spending money in disease care, where about $4 trillion a year is spent on the treatment of disease, and very little is actually spent on keeping people out of disease. I've been in this industry for about 15 years. I've worked extensively within the medical world with physicians in areas such as helping people deal with addiction around tobacco, around general health and wellness, weight, etc. And for the most part, a lot of physicians I've worked with are pretty cynical about the idea that one can actually reverse using lifestyle interventions. And in fact, the evidence would show that that's true. So what Airvale is really focused in on is how do we create a system for an individual that uses the best data that we can develop on a personalized basis, use that data in combination with the right type of lifestyle interventions and coaching, and then measure change over time to see whether or not we can actually take someone from a state of less well to more well. This is what Lee coined the term scientific wellness, and Airvale is the manifestation of that. So Airvale launched as a commercial entity for a wide number of reasons, but its primary focus has been to take this P4 medicine paradigm and bring it out and then grow it and scale it. The idea behind it is to start with a multi-omic analysis that uses 4 quadrants of data. First, we do whole genome sequencing on each individual. Right now, we're primarily working with several dozen SNPs that we know to have very wide, well-understood gene-bioenvironment interactions, but simultaneously, our research organization is developing a correlations network that is separating signal from noise to find correlations that we believe over time could be researched and studied for novel approaches on multi-omic analysis that allow us to tell more about a person's, not just their wellness, but their transition toward disease. So whole genome sequencing to start. In addition, we do a wide number of clinical assays to look into the blood to understand how a person is transitioning towards disease or reversing or mitigating that transition. And I'll give you some examples about how we prioritize and do that, which dimensions we look at. But we do look at a wide number of clinical assays to relate them back where relevant to genetic interactions, to put the genetics in context and to put the clinical chemistries in context. Not to come up with definitive solutions, but to provide some level of context where relevant. The third area that we gather an extensive amount of data is looking at lifestyle data. We do that by taking all of our participants in our program use a Fitbit, many of them on a 24/7 basis, where we're getting activity data, we're getting sleep data, we're getting resting heart rate data coming in every day simultaneously. Dr. Cobb's there, you see, rocking is it your Fitbit there, or what do you got going on there?
Yeah, getting recorded
As we speak, good. We can see what your stress levels and resting heart rate are later. Yeah. We also we also look at assessment data that we gather at various times to understand qualitative components of an individual's life. The fourth quadrant in what we do is we look at certain discovery assays. So we're doing ELISA-based proteomics in the area of inflammation and cardiovascular risk right now. We're also looking at broad-scale metabolomics. In addition, we also do microbiome 16S rRNA sequencing, which is a fairly basic level of microbiome genetic sequencing right now, but giving us some understanding of indication of understanding the interactions on microbiome. Now, a lot of that data then is reviewed. We have relationships with outside external physicians that do an initial review of the data to find any medical concerns. Airvale does not diagnose. If we see something that is indicative of disease, and of the about 1,200 people we've enrolled in the program, say approximately 5% of them have come in with frank disease that was missed and not diagnosed. In all those cases, we turn them right back over to their primary care physician with the data, and a diagnosis takes place. Common examples of disease that we find are type 2 diabetes. Another common one is anemia. And a third one that we've actually found a fairly not a wide range percentage, but nonetheless seen a percentage of is hemochromatosis, where we see people homozygous for the variants as well as having incredibly high ferritin levels undiagnosed. And in those cases, we turn them immediately over to their physician for treatment. We consider ourselves a healthcare alliance. We work very closely in many cases with our participants, who we call pioneers, as well as with their physicians, to share data and make sure that the physicians are getting the data they need in order if there's any medical treatment. We do not interfere in any way, shape, or form with any medication treatments, but we have been able, through lifestyle interventions, to be able to see clinical changes over time that the physicians have used to make modifications around choices on medication, particularly on reducing people's use of medications around type 2 diabetes. Once all this data is gathered, as I said, it's not only reviewed by physicians, it's then brought into a software platform that we've built. And the platform does two things. One, it creates a very easy to understand, direct sort of view of this data, that which is actionable for a participant in our program. That participant is paired with a dedicated nutritionist who is a registered dietitian, many of whom have come through a background of integrated functional study. And then those coaches help them do three things. One is to help look at this massive data and find that which is most actionable, the best and most likely opportunity to improve one's wellness. Two, to take those and determine out of that actionability, what is somebody willing to do? What are they willing to change? Most of it in the areas of nutrition, activity, some very mild supplementation where we have good clinical evidence that the supplementation can work. And then the last component of what we do is we hold people accountable in a positive way towards making lifestyle change. This personal coaching is something that we've used extensively in other areas. My last company, Free and Clear, is the largest provider of tobacco cessation counseling and services. We serviced about 1,000 people a day who proactively enrolled in our program, had a quit rate that was 6 to 8 times better than what you would find in most other just medication-only approaches. Throughout the year, we serviced about 400,000 people per year and had a quit rate that was measured response, an intent to treat rate of about 20% and a response rate of about 40%, substantially higher than what you would get from other areas. That was very much tied to the relationship that our coaches had in addition to medication support and using common components like cognitive behavioral therapy. What we're fundamentally doing with each of the individuals in our program is we help them understand that for most of them, no one wakes up in the morning and says, today's the day I'm going to be healthy. That's an abstract. What we instead do is take all this clinical data that we've developed, this genetic data, this view of them, and we tie it to core motivation. We really work with each individual to understand that which drives them, that which they find joy in, that which they are willing to anchor change around, and use that as the basis for change. We have many people. A common example I use would be someone who came in, for example, with a wide range of cardiovascular risk factors, well engaged with his primary care physician. None of this data was new. He was unwilling to make change. But what we were able to identify with this particular individual was that he was an avid nature photographer. However, he was no longer able to go to the areas in which he wanted to go up in the mountains to take photos because of his weight and knee problems. So we anchored his behavior change on that desire to be able to go into the mountains and make a change. That goal set allowed him to lose about 30 pounds over about 6 months, reverse his cardiovascular risk factors simultaneously, but that was what really wasn't a side effect for him. What the real effect was his ability to do that which mattered to him. So to give you some context, we've built this company. Where did it start, and why did we choose to go down the route of taking this multi-omic approach and then marrying this behavior change into the model? Starting in 2014, the Institute for Systems Biology launched a study under an IRP where we looked and said we're going to take 108 individuals and see if we could do 4 things with them. One is, could we logistically gather all this data? 2, once we gathered all this data, could we separate signal from noise and find meaningful recommendations? 3, could we actually coach to those recommendations? And 4, could we see actual clinical changes in people's health once they went through this program? The results of all 4 of those were positive. After about 10 months in the program, we were able to take people, most of whom came in common baseline, average American lifestyle, high risk for 60% were overweight or obese, had a wide range of cardiovascular risk factors, pre-diabetes factors, able to take them through a series of lifestyle changes, work with them to understand where there were heavy gene-by-environment interactions, use that genetic information in context to the clinical data, looked at the microbiome as a series of abundance and diversity scores to talk about vegetable intake versus meat intake, used the lifestyle data to look at people's sleep patterns, the activity that they were doing, gathered all that data and created a series of fairly concise recommendations that our coaching organization was then able to work with. The results were substantially statistically significant and clinically significant changes in individuals' health over about 10 months. These 108 individuals, we call them our original pioneers in the program. We saw that there were enough good results that we felt it was right time to roll it out commercially and start small. So we've been gathering information, building out our program, building out our software platform. We went from about 100 people finishing up the program in early 2015, launched the company in July 2015, and now have enrolled about 1,200 people directly into our program. What we were able to find was that we were able to get a lot of people to engage in behavior change using their genetic data and their clinical chemistries in context. It became a powerful tool, but it wasn't just what they looked at. They looked at a broader range of topics and used this data in context. So where are we now? Company's been launched and we're in the process of growing the business. So we've really been able to align the business for individuals on 4 very specific deliverables that we're able to take all this data and create extreme personalization, help them understand more about themselves than they've ever done before. A lot of people coming into our program have been fairly frustrated with their inability to understand more about themselves, to be able to engage in a deeper discussion on their own personal health, and they want to own that. So we're able to create that model and that methodology for them to do that, to integrate their view of their health, to create actionable recommendations, and then to use the coaching relationship to create relationship-based accountability. We've now taken our information and really created a concise view of that data. When individuals come in and they fill out the assays, the assessments, they do their assays, we gather this data and we now put them into one of 6 prioritized health dimensions: cardiovascular risk, prediabetes or diabetes risk, general inflammation, optimal nutrition, looking at healthy aging, which really is oxidative stress. And the last we look at is we do a cortisol and DHEA stress test and over a 4-point test, and we look to see how they rank in terms of stress. They're put in one of 6 dimensions. One is prioritized and a secondary one is established, and then lifestyle recommendations are built up around those 6 dimensions and the goal to reverse them. We've built out our software platform. We've built out a mobile app to allow them to engage in behavior change. And I'll just get to the results. We've also built out a scientific advisory board that's Dr. Lee Hood, who's actually now the Chief Science Officer here at Providence, is working with us directly on, and that scientific advisory board has really created a mechanism by which we can continue to advance the science in this area and to really make sure what we're doing is really focused on the areas of scientific wellness. Along those lines, we're actually just we've submitted and expect to be published in our first peer-reviewed journal quite shortly, looking at the results from the Pioneer 108, and we'll continue to do peer-reviewed papers and publish off of that. So what do our clinical results look to date? What we found is when we box our folks into coming into the program with high, medium, and low risk factors, and then looking at substantial relevant biomarkers, we're able to see that we are trending everybody into the right direction for the most part. Definitely outliers, but when we look at folks who fall into our high baseline that came in with a high baseline fasting insulin, elevated or normal, we can see that we're trending them all towards relevant changes in LDL cholesterol, vitamin D, homocysteine as a measure of cardiovascular health. In all cases, we're trending the folks who are highest down to areas where they would be either elevated or normal, folks from elevated to normal, and those within normal staying within normal. We do this not through just the data itself, but through the engagement patterns. Interestingly enough, I've been involved with this industry, as I said, for many, many years. Our scientific advisory board just met a few months ago, looked at our data on our engagement, and were shocked about the level of engagement and the length of engagement we're able to keep people. 12 months into the program, people are taking upwards to 85 to 90% of our folks are actively engaged, measured by still taking and getting their blood drawn, filling out other assays, and taking at least monthly phone calls with their coach. Last, what I want to finish you all is how is this data going into our research and what are we doing with the research? So in addition to getting individualized and group and aggregated clinical results, we're also building out a multi-omics correlation network. This correlation network started with 108, is now at about 1,200, and what we're doing there is taking creating the ability to separate signal from noise and find statistically significant, non-spurious correlations between multi-omic analyses that allow us to see trend towards certain diseases. I'll give some specifics in a moment. To date, we found approximately 4,000 statistically significant correlations between 6 data types. We've been able to find that the top existing correlations are between existing disease treatments, particularly in areas of cardiovascular and diabetes risk. In essence, what we're doing now with the correlations is identifying a series of positive controls, i.e. we find correlations that we find to be interesting, we search the data, we search the existing research, and we find other examples of where this research was found. So that data is now starting to build up for us to be able to go and build the model. We're also finding some interesting novel correlations that are early. One example of a known correlation, for example, we found was an association with within our community analysis around cardiometabolic community, and by looking at certain proteins, for example, FGF21, and then finding those relationships between that and other cardiovascular risk factors. That we identified via our proteomics, clinical data, and genetic data, found that relationship, and then we're able to actually find an existing 2 studies fairly well established that were done many years ago in the pharmaceutical community in this area. So not only are we finding these examples of positive controls, but we're also able to now find interesting novel correlations. So for example, one of the areas that we found particularly interesting is that we're finding novel relationships between certain dynamic biomarkers such as changes in cysteine levels, and one's genetic risk for Crohn's disease. So as we see the number of genetic markers for Crohn's disease go up, we're finding a dynamic relationship with the amount of cysteine in the blood, and able to actually start to measure that, and over time start to look to see whether or not that might be a predictor of a tip towards that disease. So our goals with all of this data is, because we're looking at this data frequently, for each person in the program we're drawing blood at least twice a year. We just had approximately 60% of our initial year 1 enrollments folks have re-enrolled into year 2. So now we're getting 3, 4, 5, 6. In many cases, we have folks from our Pioneer 100 who are 3 years into the program. We're getting views of their clinical and discovery data, proteomics and metabolomics, over a longer period of time. As that data volume grows, the statistical significance of what we're finding grows, and the opportunity to find novel correlations in these multi-omic networks continue to grow. The last slide I'll finish up with is to tell you that in addition to doing the work that we do at Airvale, we've actually now just launched our first series of co-partnerships within the Providence system. So ISB, Airvale, and Providence we haven't formally announced this yet, but you'll be hearing about it shortly. We just announced our first portal project, which is 2,000 Providence employees in a 3-year scientific wellness study, where we'll take these employees through a 3-year Airvale program with the goal of measuring changes in healthcare utilization and healthcare cost over that 3 years. In addition to that study, which is now under IRB review, we expect to get it launched this quarter, starting in Renton and then in Seattle, and then going down to Portland in California. We then expect what will happen is we'll be developing other areas of research where the Airvale type analysis, in addition to ISB's expertise and Providence's clinical expertise, can come together. 3 portals that have been established that we're looking at right now include breast cancer survivorship, the ability to take an individual who's survived breast cancer from survive to thrive, what that would look like. A second area would be diabetes. A third area would be glioblastoma, which Dr. Cobbs is involved with. And last is an opportunity to look at Alzheimer's and look at the ability to recognize early cognitive decline, see that happening, and look at certain lifestyle interventions that are being done out of UCSD. Dr. Dale Bresnan is doing some really fascinating work in this area, and the potential to measure cognitive decline and change in a multi-omic system for the potential to be able to identify and either reverse, mitigate, or arrest early cognitive decline before it actually transitions. So those are other portal areas that we're working on right now within the Providence system. Airvale itself will continue to grow with its direct enrollment to build out its scientific wellness and understanding. We've now launching in California, Oregon, and Washington, and we'll be expanding state by state as regulatory allows. So that's been about roughly 15 minutes of discussion. I think you've got about a few minutes left in this. I'll be happy to answer any questions about what we do.